hey AP, hope you're doing well when you watch this video. All right, so this is part 1 of gene regulation. There's going to be 2 parts. And so we're doing the notes at home. Yay! It's going to be awesome. It's going to be so great. And then we're going to talk about them on Tuesday and, of course, do our, modeling activity with operon noodles and stuff. So that'll be pretty fun. But yeah, this is we're going to start off talking about alcohol. Also, an interesting topic. And so from an old textbook, I'm not going to read you the entire story, but basically what happened was they discussed alcoholism and how alcoholism can be genetic. And so at the James Murphy at Indiana University has bred a genetic strain of alcoholic rats called P rats. So I want you to listen to this. P rats show many of the symptoms of addiction, including compulsive drinking, tolerance, and withdrawal. These rats appear more anxious than wild-type rats. Again, what does wild-type mean? Spending more time in a closed rather than open environment. Drinking alcohol alters this behavior and seems to relieve their anxiety. There may be a link between a particular protein and alcohol consumption. It's called CREB, and I want to get you the introduction. Like, why does this matter? Why does this conversation matter? This is why. CREB is abundant in the brain and regulates the expressions of hundreds of genes that are important in metabolism. CREB becomes activated when it is phosphorylated by a protein kinase. Again, hopefully all you know this protein kinase A, which is turned activated by secondary messenger cyclic AMP, CAMP. In an effort to understand the molecular basis, another neuroscientist at another university compared CREB levels in brains of P rats, CREB levels in the brains of P rats, and wild-type rats. They found that P rats have inherently lower levels of CREB in their parts of the brain. When these rats consumed alcohol, the total levels of CREB did not increase, but the levels of phosphorylated CREB did. It is the phosphorylated version of CREB that regulates gene transcription. So why does this matter? Why is this important? You're kind of like, well, what is all this CREB stuff? And I'm going to show you some graphs here. The prospect that CREB, a molecule that regulates gene expression, is a key element in the genetic propensity for alcoholism is important because it begins to explain the molecular nature of a complex behavioral disease. Nature and nurture, right? What you're born with and how you live. And so that came from this book right here. Just a little disclaimer. And so yeah, let's talk about, first off, let's talk about the objectives here. And so objectives for this lesson is all about, regulating gene expression. So regulating gene expression, like right here. Okay, and so what we're doing here is we are, maybe I can highlight instead. That'd be probably better. So what we're doing here is we are talking about regulatory sequences are stretches of DNA that interact with regulatory proteins to control transcription. Okay, and so epigenetic changes can affect gene expression through reversible modifications of DNA or histones. The phenotype of a cell or organism is determined by the combination of genes that are expressed and the levels at which they are expressed. Observable cell differentiation, which means our skin cells are different than our muscular cells, results from the expression of genes, right? So if our cells have the entire genome, but they don't express all the genes, then genes have to be able to be turned on or off, read or not read. Induction of transcription factors during development results in sequential gene expression during development. Explain how when you're developing, how does certain cells go into a certain lineage, maybe into the muscular system versus the skeletal system? Well, that's different factors being around, transcription factors. Explain how the location of regulatory sequences relates to their function. So this is both prokaryotes and eukaryotes have differences. I need to do a different color. Differences when it comes to how they regulate genes. In prokaryotes, we're going to talk about operons a lot in this video. The lac operon versus trip operons. In eukaryotes, they're influenced by transcription factors that regulate expression. So if these transcription factors aren't around, RNA polymerase doesn't transcribe the gene. Differences in the expression of genes account for some of the phenotypic differences between organisms. Again, same enduring understanding. Different learning objectives, though. Explain how the binding of transcription factors to promoter regions affects gene expression and or the phenotype of the organism. So that's probably going to be part 2, talking about promoters, negative regulatory molecules. And then explain how the connection between the regulation of a gene and phenotypic differences of cells, how do they relate? Gene regulation results in differential gene expression influences cell products and function. Okay, so that is kind of what we're kind of talking about, a little bit today. And so prokaryotic and eukaryotes alter gene expression in response to different environmental conditions. And understanding this is super important. And so here's some objectives. You can read over those for time. I'm not going to. Of course, we just read over the objectives from AP Classroom. So alcoholism. Yes, if we were in class, I would show you a couple of videos. But is there a link between a protein and alcoholism? And how does CREB regulate the expression of this? So here's an example of... Sorry, let me adjust a few things. Here's an example of what I was talking about. So in this experiment, again, adolescent alcohol exposure epigenetically regulates CREB signaling in the adult amygdala. Well, so the amygdala is kind of like your fear center, anxiety, and all that kind of starts there. And epigenetics, we talked about that. So we expose male rats to adolescents, intermittent, so drinking when they're young, or saline, which of course is not alcohol, during postnatal days, that's kind of like their teenage days, and evaluated the regulation of CREB dynamics in the adult amygdala. A subset of these adult rats was exposed to an acute ethanol challenge. AIE, alcoholic, decreased CREB, phosphorylated CREB, and changed these protein levels in adult amygdaloid brain structures. Okay, you're like, well, why does this matter? Well, let's go on to the next one. So here's the graph here. And so the graph shows that you can see the adult amygdala, CREB, immunolabeling. Here's saline, and here's alcohol. Okay? And so you can see right here, CREB, mRNA expression. You can see alcohol lowered CREB. Statistically, right? Look at the error bars. Right here. mRNA expression completely altered it for CREB. Okay? So when you drink alcohol as adolescents, you lower the expression of CREB when you're an adult. Why does this matter? Well, right here, in conclusion, and so why does this matter? So you can see right here that adolescent consumption of alcohol causes lasting changes to the CREB signaling pathway in the amygdala. Again, like an emotional center, anxiety, fear, has a lot to play with the amygdala. Decreasing all these levels. This is paralleled by a decrease in CREB1, and that returns to control-like levels after acute challenge of ethanol in adulthood. Collectively, the decreased levels of signaling molecules in the CREB pathway seen in this study, along with the increased anxiety and alcohol preference previously reported in adolescent ethanol adult animals, add to the growing body of evidence that these levels in the amygdala are negatively correlated with anxiety-like and alcoholic drinking behaviors. So what they're saying is when these levels are low, anxiety and alcoholic drinking behaviors as an adult is high. So your choices right now matter. That's why this is so important. Your choices right now matter. How you live your life. And so this is just another example of seeing how this is affected. So anxiety and alcohol abuse disorders, a common role. It's been hypothesized that anxiety disorders play an important role. Right? Here's CREB again. Propose a CAMP response element binding protein has a role in anxiety and alcohol drinking behaviors. The CREB gene transcription factor regulates the expression of neuropeptide Y, and decreased concentrations of neuropeptide Y are implicated in anxiety. Look, right there. We won't read all of it, but decreased levels, concentrations are promoted in anxiety and alcoholic behaviors as an adult. So proteins, transcription factors, epigenetics, genes being turned on or off, even how you live right now, it all has a play. Okay? So a molecule that regulates gene expression is important because it begins to understand the complex disease. Okay, so this is from another textbook, Anablebs, commonly known as Cuatro Ojos, 4 eyes, glides in freshwater. But here's what's crazy. One eye, the eye's upper half is well suited for aerial vision, and the lower half is for aquatic vision. You have to have different proteins to do that. So this is specialization. Different genes have to be made and products made. And so the cells of the two parts of the eye express slightly different set of genes involved in vision. That's crazy to think about how in one eye you have certain proteins on, certain proteins not on. And so gene expression, what is it? It's the process by which information encoded in the DNA directs the synthesis of proteins, or in some cases, RNAs are not translated. So there's 3 different ways. We've already talked about them in many ways, these 3 different ways. Signal cascade pathways, cyclins in the cell, viruses, they hijack expression machinery. I'll review that in lesson 2. And so this is, gene expression has 3 different places it can happen. Before, during transcription, before, during translation, and after translation. So here's a picture here. We can control if the gene is transcribed. We can control if the RNA actually makes it to the ribosome. We can degrade it. We can alter the protein possibly. So there's a lot of ways to regulate. And so gene expression begins at the promoter region, which is where RNA polymerase binds. Regulatory proteins are called transcription factors. So there's repressors, which of course prevent transcription, and there's activators. And all these are, transcription factors. They're proteins, okay? And so here's an example of that, negative binding, no transcription. RNA polymerase can't bind. But maybe RNA polymerase does need this protein around, so if it's not around, it does bind. And so bacterial cells, what do they need to survive and reproduce? Go ahead and jot a couple of thoughts down. Think about it, at least in your mind. What do cells need to survive and reproduce? One of the things that they need is they need to be able to, break down, break down products that are around, make products that aren't around, regulate energy. We don't just need to be wasting energy here. And so natural selection has favored bacteria that produce only gene products needed. Why do you want to make something that you don't need? The answer is you don't, right? That's not that you shouldn't. And so this is where we get to an operon. An operon, or a single promoter, serves as a functionally related genes, a set, right? So these genes are controlled by a single on or off switch. And so the regulatory switch is a segment of DNA called an operator. And I'll show you pictures with all this. This is what we're going to be modeling in class. An operon is the entire stretch of DNA that includes the operator, the promoter, and the genes that they control. Okay, so promoter region, where RNA polymerase first attaches. These are just great note cards to make. I know not many of you make note cards, but great note cards to make. Operator region is the DNA sequence where active repressor binds. Active is the key word there. Right there. And then structural genes. There's several genes coding for enzymes of a certain metabolic pathway. Metabolic could be building or breaking down. So this is a long segment of DNA, and we're talking about prokaryotes here. This is not really eukaryotes. We're talking about prokaryotes, hence why it's highlighted. And so, for example, they conserve energy and resource by making certain proteins only when they are needed. An example is lactose catabolism. What is catabolic reactions? What does that mean? Hopefully you thought, okay, we're breaking down. And then we're going to talk about a tripoperon, which builds tryptophan. So here you go. We're going to talk about 2 different operons here. We're going to talk about lactose, which of course is in dairy, and then glucose, where glucose and fructose is also going to have a little play today as well. So here's what lactose looks like. And so there's 2 different operon systems. There's a repressible operon, okay? You just need to be able to contrast these. These are involved in anabolic pathways, which means they build. This is like a tripoperon. Negative control, right? So we're building something unless we don't need it. An inducible operon is a catabolic pathway, which means we're breaking something down. It requires an inducer, which means to turn on the operon, and it's positive control. Okay, we need this on because this is around and we need to use it. Okay, and so an inducible operon is usually off, and a molecule called an inducer inactivates the repressor and turns on transcription. The lac operon is an inducible operon, so that you got to know that. The lac repressor is active, okay, which means, it's off, right? It currently shuts off. And a molecule called the inducer inactivates the repressor, thereby turning the operon on. And I'm going to show you a picture here in a second. You don't need to know this. These are the different enzymes, so I'm not even going to waste time with this. But basically what they showed was that these enzymes are necessary in breaking down lactose. And so what they measured here is okay, we add lactose, and the level of this protein goes up. The level of the mRNA goes up, and when you remove lactose, the level goes down to the baseline level. And so something is being turned on and something is being turned off as far as expression goes. And so here are them both together. So you add lactose. Obviously, the cell responded to the environment. You have to be able to respond to the environment to be alive. And so that is important. So at what level is the response to lactose according to these graphs? Transcriptional, translational, or post-translational? Well, hopefully you can see right here that the induced levels stay pretty high, the protein levels stay pretty high, but what dropped? Well, the mRNA dropped. And so we are transcriptionally regulating this. We are letting the gene be turned on or off right from the get-go, which is the best way to conserve energy, because why would you make a protein and then have to break it down? That's a little bit more complex, usually meant for eukaryotes and stuff. And so the response to bacteria is at the level of transcription. Genes are expressed most of the time, so some genes are pretty much always expressed. And so those are called constitutive genes, another key vocabulary word that you need to know about. Okay? So the 3 genes, you don't need to know those, but here's the promoter region. Here's the, for the, gene I. Here's the gene for the repressor protein. Okay, so those are related. And here's the operon, promoter, operator, and then the gene. So this is an operon. Promoter, operator, genes. The operator is where the repressor binds. Okay, so let's look at some pictures here. Only in prokaryotes, super important, not in anything else. Crazy, crazy important with that. Okay, so regulatory sequences are here, which means these are what turns the genes on or off, and structural genes are making enzymes, proteins, things we need for this metabolic pathway. Operator and promoter. So the operon includes an operator, we talked about that, and this is where the repressor binds. Inducible operons are turned off unless needed, super important, obviously it's a blank. Repressible operons are turned on if not needed. Have to know this. You have to know this. Inducible operons are off unless the molecule's around that we need to break down. Repressible operons are on. We're making a crucial product unless we can get that product from the environment, and then we don't want to waste energy. So lac operon is inducible. Let me show you the picture. So here we go. Here's your active repressor being made from this gene. It's active. Constitutive gene, pretty much. Making that protein, it's binding to the DNA right here. Non-covalently, of course, due to bonding. RNA polymerase can't bind. These genes are not on when lactose is absent. But when lactose is present, lactose, a form of lactose, actually acts as the inducer. It binds to the repressor, thereby inactivating it. Shape leads to what? Hopefully you said function. And so RNA polymerase then is able to bind to the promoter region, and 5 to 3, right? 5 to 3. Oh, my bad. I don't know what's happening here. Oh, wow. You can control zooming out and zooming in. I did not know that. Hey, learn something. But this is description of transdirection. 5 to 3, right? It's building 5 to 3. And so these genes are transcribed together. That's an operon. So here's your proteins that are necessary. You don't need to know these names, but these are proteins necessary for the breakdown of lactose, which is energy. Lactose is a sugar. We need to use that if it's around. And so I just explained all this. You can read this over in class. So here's just another picture here. You can see the repressor protein is actively, and you'll kind of model this, right? We're going to model this, and you're going to explain it in your own words. Maybe creating a fictional operon system, breaking down another product. But one of you are going to build an inducible one, and one of you are going to build a repressible one. And so you can see it's active for the repressor. And so it's binding to the operator region, blocking transcription. Got to know that. RNA polymerase cannot bind. Okay, let's zoom in a little bit. Can I zoom in? Oh, I can't zoom in. I lost the ability to do that. So you have your start codon, stop codon, and these are all, look, 3 different proteins, right? Lactose, allolactose, is the inducer, which means it binds to the repressor protein, inactivates it due to shape change, conformational shape change, and therefore RNA polymerase can read it. This is a way of regulating a metabolic pathway. And all of this is examples kind of feedback, inhibition. With tryptophan is an amino acid. With tryptophan, you got to make tryptophan. But regulation of gene expression, if tryptophan's around, if it can act as a repressor somehow and shut this process down, if we can shut this process down here, we save energy if tryptophan is in the environment. Okay, so tryptophan is another one. And so here's the operon's the basic concept. The operon can be switched off by a protein called a repressor. So this is the trip operon, basically. And And so, yes. And so we're going into the trip operon now. And so the repressor binds to the operator and blocks the attachment. The repressor is the product of a separate regulatory gene, which is not part of the operon. Okay, so that's kind of basically what we were talking about there. Okay, so, oh, my bad, delay. So the repressor is active in an inactive form depending upon the presence of other molecules. A co-repressor is a molecule that cooperates with a repressor protein. So tryptophan operon is an example of a repressible one. So it's on. We're making tryptophan. Why would we ever turn it off? Tryptophan is a necessary amino acid. Because if it's present, the cell can stop making enzymes for its synthesis. It's all about homeostasis. So it binds to the repressor, causing the repressor to bind to the operator. Okay, so pictures again are crucial, important, but basically it's on and they're transcribed. These enzymes are made, we're making tryptophan, necessary amino acid. But if tryptophan is present in adequate quantities, it binds to the repressor protein, thus turning the operon off. In this repressible operon, the repressor is active only in the presence of its co-repressor, tryptophan. So it's turned off if tryptophan levels are high, again, responding to the environment. So here's what happens when tryptophan is absent. Inactive repressor, right? We're still making constitutive gene. We're making the protein, but RNA polymerase is binding to the promoter region. We're, transcription is happening. We're making all these enzymes that are necessary to build tryptophan. But if tryptophan is present, it is a co-repressor, which means it binds the repressor protein, activates it, thereby allowing the protein to bind right here to the operator region, and thereby RNA polymerase cannot bind and transcription does not occur. And so this is an example of an operon, inducible and repressible. Transcription prokaryotes can also be regulated by activator proteins. Activators can regulate both inducible and repressible systems. Many genes are controlled by more than one regulatory mechanism. So yeah, we're just, of course, scratching the surface here, right? This is not, end-all, be-all situation. An example of that, a positive gene regulation, is E. coli. Actually, there was a couple of questions one year on a test and about this, and again, it's like a couple of slides, so it's interesting how questions are made. But E. coli will preferentially use glucose when it's available. So yes, why do we want to use lactose? If we have to break it down, lactose is a disaccharide. And so why would we want to use lactose if we can just use glucose? So when glucose is scarce, CRP, CAMP receptor protein, acts as an activator of transcription. And so it's activated by binding with cyclic AMP. So activated CRP attaches to the promoter region of the lac operon and increases the affinity of RNA polymerase. All right, so we'll continue on. Sorry, I was interrupted by a certain student. So CRP is activated by binding CAMP, right? Okay, so why is this? Why are we adding on to this? Because activated CRP, again, regulation, yes, attaches to the promoter of the lac operon and increases the affinity of RNA polymerase. So this also increases the likelihood that we are going to transcribe genes necessary for the lac operon. Okay? So when glucose is low, this protein is activated and it increases transcription for lactose genes. When glucose levels increase, of course, CAMP levels fall off because, we are using all that up, in other metabolic pathways. So CRP detaches and transcription proceeds at a very low rate, even if lactose is present. So this is just another example of another factor around necessary to help out. So here's a picture of this. All right, so CRP binding site right here. So when lactose is present, glucose is scarce, CAMP levels are high. CAMP binds to it, activates it, boom. But when glucose, where's CAMP? CAMP's nowhere to be found because glucose is everywhere. So this is inactive. And so even if lactose is around, RNA polymerase is less likely to bind because this increases the chance of it binding and transcribing. So again, just another way of doing that. And how does all this work? Oh man, allosteric regulation, super important. And so here's just a picture of that. And here's some questions for you. So I gave them in your notes. Please do them. We'll talk about the answers in class, hopefully, before we kind of model, although that'll be debatable. So, nonetheless, I hope you enjoyed this video. I'm going to do like the webcam right now to be extra weird. I don't know. but it's good stuff. We're gonna make it. I'm so proud of you. Keep up the good work. Love in Christ.